you for coming along. Thank you for having me. It's good to be able to do these type of talks. And yeah, if it's too much and need some clarification, please hold me to account. I need to maybe improve my outreach. So we'll give this a go. So I'd just like to give a brief introduction to my background. So as Susan said, I'm purely scientific, purely research. I did an undergraduate in biochemistry from the University of Glasgow, then moved over to Edinburgh, where I did a master's in drug discovery and translational biology, and then a PhD, which I finished last year, which was in the broad theme of genetics and molecular medicine. And I'm now working still with the MRC and genetics unit as a postdoctoral fellow, and very much focused on polycystic liver disease. So my scientific interests are in the cell and molecular biology of human disease, essentially, and this is very much focused to the liver and to polycystic liver disease.
Scott, is it possible you could maybe change your angle slightly for the mic? You're getting an echo through, and someone has said that they're struggling with the quality.
Okay.
Now, it might be their device. I don't know. Is anybody else struggling with quality there?
Well, it could be me.
Jenny, is that a thumbs up? It's okay? Perfect. We're getting a thumbs up there from a couple. I can see of faces on the screen. It might be that person's device as well, so maybe playing up, maybe not with the best connection.
Okay.
Keep
me right.
Sorry, just to say, clarify, it isn't that clear, Scott, at your end.
Right.
A little bit muffled. So if it is possible to, sorry, that was a misunderstanding of thumbs versus thumbs.
Yeah.
What I might do is I may join in my phone if that's maybe easier.
Entirely up to yourself. I'm happy to bring up your slides. I have got a copy available.
I think I'll stay on the computer, but maybe choose the phone for sound. That's going to be an issue.
You can certainly try it. You may find you might get an echo back or a delay depending on the internet speeds.
That's true. Does it sound any different to anyone now if I speak louder, or I don't know if it's my...
Jenny, could you perhaps unmute and give us your view there, please?
Yeah, the difference between your voice, Susan, and Scott's is quite clear. It's very, yours is very clear. Scott's is quite muffled. I don't know if that's what others are experiencing, but sorry to be the bearer of bad news. Sorry.
We'd rather sort it out as against everyone's time. Got iPhones just joining us.
Okay.
That's not getting any better at this, am I?
We had I'll just say a rather large echo.
Yeah, I heard that.
Yeah, but you're actually, it's fine now, Scott.
Are you sure?
Honestly, Jenny, you're going to be the judge there.
Yeah, that's fine. I think if you just speak a bit louder, it gets clearer.
Okay, well, I will go loud, and if it is an issue, I think I can make the phone work, but maybe not for now. Okay. Back to where I was. Just interrupt if it's muffled again. So I was about to give an introduction to the liver and what its job is. So we might be familiar with this, but just to give everyone the same background. So the liver is the largest internal organ in our body, and it has many different jobs. So this diagram on the right is showing where the liver sits. So it's on the right side of the body, and it plays a role in digestion, energy storage. So when we run out of energy, we'll use energy that's stored in the liver. And finally, it's used to break down drugs. So, alcohol, paracetamol, that type of thing that you might be familiar with. So that's common roles of the liver. And in addition to the main part of the liver, we also have the bile ducts. So here's your liver shown in grey this time. And we have these biological tubes that are shown in blue in this diagram. So they go all the way from the top of the liver and feed into the gallbladder shown here, and then also down in towards the intestines. So what their job is to transport bile. So the bile is made by the grey part of the liver, and then transports down towards the gallbladder or the intestine. And the role of bile is to help digest fat from the food that we intake. And this is an image from one of my colleagues here in Edinburgh, where we've actually got the bile ducts of a mouse liver shown here in blue. So we call this a biliary tree, and you can imagine why. So it's a bit like tree branches. And you can see that they kind of get a bit smaller at the top, and then they gradually get larger as they pull towards the intestines of the gallbladder. So that's the background on the liver. And I thought I'd just give, a kind of basic introduction to polycystic liver disease. So we characterize polycystic liver disease as a liver that has multiple cysts, and by multiple we mean 10 or more. And the reason why I introduced the bile ducts to you is that the cysts of a polycystic liver are made of the same types of cells that make the bile ducts. And these cysts get larger with as a patient gets older, and they fill up like water balloons, which can also be quite painful for the patient as the cysts start to push on, the stomach or other organs within the body. So what can we do to treat polycystic liver disease? So the only available treatment options in the UK and globally typically involve surgery or procedures in the clinic. And there's different things that you may be familiar with, for example, resection. So if we find that a lot of the cysts exist in one part of the liver, we can, or I say we, I'm not a surgeon, but a surgeon can remove part of the liver, and this liver will grow back. So we know that you can remove up to 50, maybe even 60% of a liver, and it will grow back to its original size. However, if there are cysts all throughout the liver and it's causing quite a lot of issues, the ideal treatment would be transplant. But what about medication? Unfortunately, there are no medications that are approved in the UK or globally to treat polycystic liver disease. However, there have been one family of medicines that have shown promising results in stopping these cysts getting larger in clinical trials. And these are called somatostatin analogs. Again, you may have heard of these. But we really need to try and find other medications that can work to stop these cysts from getting bigger and to improve the quality of life for patients. So my overall research aims are to understand more about the biology of polycystic liver disease, and then to use this understanding to find ways that we can treat polycystic liver disease. So other medications that can stop cysts from getting larger or to stop new cysts forming. So just going back again into a bit more of my background, this is the front cover of my thesis, which I'm not going to go into too much detail, but I was focused on cell biology and, introducing some concepts that you may have heard, in high school biology. You may have even heard some of them more recently from being part of the PKD community. But here's a cell, and in the middle we've got this nucleus. So this contains the DNA, and we call this the brain of the cell. So it's got all the instructions that are needed to make this cell do its work. There is a cytoplasm that we call here. So this is the site of these biochemical reactions. So all the processes that are required for a cell to do its job. And then on the outside, we have this membrane that acts as a barrier and stops things from getting in that we don't want to get in. But what I focused on in my PhD was not really any of these parts of the cell, but the cilia, or cilium for one, which again, you may have heard of from other events hosted by the PKD Charity. So these are these little hair-like structures that appear on the top of a cell. And cilium is actually a word from Latin, and it means eyelash, and cilia is plural, so it means eyelashes. And you can imagine this diagram shown here is almost like an eyelash. And they are these appendages that sit on top of cells across different cells in the body, and they perform many different jobs in the body. So here are two classic examples that most people refer to. The first one are the cilia of the airways. So here we've got the windpipe or the trachea, and these cells have lots of cilia on top of their cell. And what happens when we breathe in, we sometimes breathe in microbes like bacteria and viruses or pollutants like smoke. And we don't really want those things going to the bottom of our lungs. We want to keep them away from the lungs. So these cilia will start to beat upwards, they physically move. And it's almost like creating a draft so that it stops things from getting to the bottom. And you know, if you ever have the colds, you'll make a lot more phlegm in your airways. And when you cough, you tend to bring that up. You might realize that when you cough, the natural response is to swallow straightly afterwards. So what happens is we're bringing up things we don't want to be in our airways up to the throat. After we cough, we naturally swallow, and that will get injected by the stomach. And you know, the stomach's quite a harsh environment for things like bacteria and viruses because it has lots of acid that can essentially kill them. So you can think of these cilia like the bottom of this sweeper that's sweeping off litter from the pavement. That's the same type of idea going on here. But the cilia in the liver and in the kidneys are a bit different. They don't move. What they do is they just kind of sit there quite happily, and they receive signals, chemical signals from the environment. So these could be things that tell the cell to grow or to divide into two cells. Or in the case of the liver, these cilia can sense what is in the bile. And you can think of these type of cilia as the antennas you get in an old TV or a radio, where it's sensing these signals and then feeding that down towards the TV or the cell body, so the main part of the cell. So what I did in my PhD was essentially making bile duct cells that don't have cilia. And in my PhD, we use mice for our research, and we like to be transparent about that. We're very privileged to be able to work with animal models, and mice is a good example of a model used a lot for human disease. And that's because they're mammals and we're mammals, so a lot of things are quite similar. So what I did in my PhD is, we have these bile duct cells that have a cilia in the liver. We're able to use genetic tools. We call them molecular scissors. I'm not going to go into details too much, but we're able to change the DNA within these cells, and we can then make cells that don't have cilia that work. So they have these faulty cilia. And the question we wanted to ask was, what happens to the cell as a whole after they lose the cilia? So at the very start, I showed you that nice tree-like structure, the biliary tree shown in blue, and that was a three-dimensional image. Here we have a typical mouse liver, this time only in 2 dimensions. And you can see all the pink and purple is a dye that is stained the cells. You see some white spaces, so these are largely veins and arteries. If you focus on this area, we see a normal bile duct. They're quite small, really, and it's quite hard to identify if you're not used to looking at the liver. But in the mice where we had the cells that have no cilia, you see there's quite a drastic change. So instead of being mostly pink and purple, we see a lot more white. And this time it's not all veins or arteries, there's a lot of cysts now in making up this liver. And when we count the number of cysts in these livers compared to the number of the bile ducts in these livers, we see that there's a lot more. That's what this graph is showing at the bottom. We have a lot more cysts in these livers compared to the bile ducts in these livers. And the way I've shown this graph is a bit funny, but basically what it's showing is that these are generally much larger in size compared to the normal bile duct as well. So thinking back to that blue tree I showed you at the start, here we now have a 3-dimensional image of some cysts grouped together in a liver. And you get an idea of the kind of complexity of these. They're quite complex structures. They've got a lot of depth to them, so they're quite thick. And you may recognize this image as it was on the cover letter or the front page of the newsletter recently. So that was how we were able to essentially mimic polycystic liver disease in the lab. But they want to ask, what is what is making this cyst develop and grow over time. And to explain that, I have to introduce a concept of cell communication. I'll try and go through it slowly. It's not too complicated, but if you're not used to it, it might not seem too interesting. But cell communication is a process by which cells communicate with each other. It's very self-explanatory there. But what happens is you have the signaling cell that can send a signaling molecule, which is shown in this little triangle, and this is then received by this receiving cell that had a receptor molecule. Okay, so it's quite simple so far. But the ultimate aim of this process is to stimulate a biological response. So that could be tell the cell to grow, to divide into two, to do other things. There's a lot of different signaling or different cell communication that occurs in our cells. And it's really quite a complicated field to get into. Well, that's my animation. So it's a biological response that is the ultimate aim of this. And what we found was that in these cysts, they made a particular signaling molecule, and that's called TGFB1. You don't have to worry about the name about it too much. And what we found was that TGFB1 is not present in the normal bile ducts, but is present in cysts. So it's something that we can see there's a clear distinction between these typical bile ducts and these cysts. And a lot is known about TGFB1 from other studies, and it's known to make something called a cell matrix. Now, a matrix is a very complicated group of molecules, a lot of proteins, and it acts like a scaffolding. So if you think of a scaffolding around a building, this matrix is kind of a coating of the cells within our body. But it changes depending if you look at the liver or the brain, different tissues, it all looks different. And what we found was that by having this TGFB1 present in these cysts, but not in the bile ducts, there was a change to the scaffolding. So we found there was a particular molecule called fibronectin, but you don't have to worry about the name so much. But you see here that it's at higher levels in these cysts compared to the typical bile ducts. And so what we decided to do was use that knowledge, because there's such a clear difference between the cysts here and the bile ducts here. We thought we would use a medicine that would stop this TGFB1 from working. So we use this medicine shown here for this little pill, and we're basically blocking off this response. So now we've blocked this off, the idea is that we'd stop making this scaffolding around the cell. And we found that there was lower amounts of the scaffolding when we gave the medicine compared to this no medicine, so like a placebo group. We saw there was less, and this correlated with a reduction in the size of these cysts. Again, it doesn't look too drastic in this graph, but that's because of the way that I've shown it as a bit fun. So we see that by blocking this signal, we then see that the cysts get smaller. But if you remember a couple of slides ago, I said cell communication is very complicated, and signaling can happen in lots of different directions. So actually what else happens is that these cells have these other receptor molecules that can bind to this matrix, bind to this scaffolding, and they then have their own biological response. So what we also decided to do was take a different approach, introduce a second medicine that stops these matrix receptors, and see how that affected cysts in this model, essentially. And what we found this time was it wasn't a change in the size of the cyst, but we found that there's a change in the number of the cysts. So once we give this medicine number 2, there was a decrease or a reduction in the number of cysts compared to this placebo group. So just to summarize what I've shown you from this research, we found that in the bile ducts we have these cells that have these cilia present in the tubes. What I did in my PhD was that I was able to use molecular scissors or these genetic tools that prevented cells from having the cilia. And as a result, we found that these cysts started to grow, and they started to make a new matrix, so a new scaffold around these cysts. And we think that this matrix is important for allowing the cyst to get bigger. Because if you remember from my previous slide, when we gave this medicine number 1, the size of the cyst got smaller. And then we also found that, the scaffolding matrix could be detected by another receptor molecule. And this time we found that this was responsible for allowing the cysts to increase in number, so we got multiple cysts. And when we gave this medicine number 2, we found that we were able to reduce the cyst number in our animals. So this was a group effort. There was lots of people involved in this study, and this included people based in Edinburgh, people based in the Netherlands, Italy, and Spain. And this ultimately led to this publication in a scientific journal in September. And yeah, there's a lot of people behind this research, which has been quite exciting to find that we can, at least in these animals, stop the cysts from getting bigger or becoming more numerous. That's all I'm going to speak about today. I'd just like to thank all these people that contributed to the work, and I also get funding from the Scottish Government's Chief Scientist Office to study polycystic liver disease. So I hope that wasn't too research heavy or too scientifically heavy, and I would be happy to answer any questions that you have.
Thank you, Scott. That was a very thorough explanation of a research topic. I will say, I looked at the audience numbers and no one turned off. We stayed static throughout. I think we had a couple of people dropping in and out due to connections, but you didn't lose your audience. Thank you. It was a very interesting. I hope the
sound was okay as well.
Yeah, there has been some comments come through on the chat. Most of them are not kind of questions directly for this evening. So I'll perhaps ask the audience if they have anything, if they're feeling a little shy, if they want to pop it into the chat. There's a new message just appearing there, or if anyone wants to raise their hand. To raise your hand, there's a little icon at the bottom. It's reactions and a smiley face. You click on that and expand it, you can raise your hands. So it's entirely up to yourself.
Can you hear us?
Yes, we can hear you.
Hi.
Hi.
Hi. My name's Raquel. I have a daughter with PKD and PLD. We have only found out, she's 19, she's only found out a year ago. So this is a complete new learning curve for us. Currently, we can't find any genetic tracing of it, but we're all undergoing tests. So this is really interesting for us. What does your research mean? Does that mean that the medication will be released, or does it have to go for, I presume it has to go for a testing trial period, or?
Yeah, it's a good question. So like we said at the start, my research is, very basic. It's the very first level before, you get to the next stages for even clinical trials with humans. So the next steps would be to probably do more non-human trials and maybe something a bit bigger, like, I think pigs are quite common for these types of things. It's not something
Okay.
It's what the pharmaceutical companies would probably be looking at. I will be completely honest, the type of medicines that we can give to mice are not the same that we give to humans, so we can get away with them. It sounds a bit harsh, but we get away with giving them other things that we typically wouldn't give to humans. We didn't see any side effects in these animals, but this was quite a short study. But what's quite reassuring is that even a short administration of the drugs or the medicines, these animals, we saw some of this changing. So we saw there was reduced number of cysts in one experiment and a smaller size of the cysts in the separate experiment. So in terms of next study, because these type of medicines are not something we give to humans, I think it would be starting a conversation with pharmaceutical companies to try and find what do they have that's actually already passed quite a lot of tests. And we actually are in conversation with a company out in the States that have they have a main medicine that they're interested in, not for polycystic disease right now. They are focused on other diseases, but we think this could have the same effect, a same beneficial effect in polycystic disease. And yeah, we're in competition with them, and we'd start again to do more similar things that I've shown you through today. And then once we get more feedback from them and how they see the things going, they'll start to kind of build this up for them, because they obviously would love to have their lead medicine treating lots of different things, so that'd be a success for them as well.
So would you anticipate that it would be a long time off a medication coming on the back of this?
Yeah, I mean, I guess, how do you define long? I mean, the typical the drug discovery process, in terms of you find a new medicine, and you want to get that into to help fight some disease, that probably takes 15 years or not longer. The idea that we take in our kind of research is that we find the kind of the biological molecules that are interesting, and then we try to find things that are already out there, so it saves a lot of the time. So the idea is that hopefully it wouldn't take as long as this normal process, which is 15 years plus. We can try and bring that down. But of course, it's still got to go through other stages of testing. And a lot of these things, not to sound too pessimistic, a lot of these things don't always get to the final stage. That's why it's important for us to explore all the different biological molecules out there and try and find new things of interest. So I might not be personally pushing for this exact medicine to go forward, but we keep doing research and keep starting the groundwork, so then we can reach out to these other companies that will then hopefully take it forward. And then the idea is you would back all your bets on one horse. You have quite a few going, and then you can try and follow up. So the idea is to bring the time down from this long, very long time of 15 years plus down to something a bit more manageable.
That's great. Thank you.
Thank you. Raquel, just for future reference for yourself and your daughter, you sound reasonably new to your journey in the PKD community. We have a lovely resource of recorded events on our website. Pop the link into the chat. You all should be able to see it. Educational events. It's under the support tab on the website. What I'll do for everyone who's registered for tonight, I will contact them most likely on Monday with the link where they can find these, and they'll find there's a question came into the conversation tonight there about genetics. There's recordings in genetics, etc. There's lots and lots of information there, and there's also a couple of medical facing PLD talks as well. So that's a fantastic resource that some of you may not know we have, that you might want to visit. So certainly it will circulate that with you, but it will be Monday. Now I can see Martin's got his hand raised. If you'd like to unmute yourself, Martin.
Thanks very much. I just wondered, does all the liver function work through the bile duct, or is there a different system that other functions are used for?
Yes, good question. So the liver does lots of things, a lot more than I could name off the top of my head. So the bile ducts, they don't really have well, maybe doing a disservice, but the main role is to form these tubes that act as delivery pipes of the bile to either the gallbladder or the intestines. They do have other jobs beyond that. But what we found was that when we gave the first medicine that I spoke about, we were actually able to saw a rescue. So from these cysts forming, the liver function was not optimal. It wasn't, the mice were fine. It wasn't optimal. But once we gave this first medicine, it managed to make the function of the liver a bit better. So, that was quite promising from our perspective. I realize I waffle quite a lot, so please feel free to chip in if I'm going on a tangent. Yeah, we think even despite that, even in patients, it's in humans, it's shown that even with these big cysts in the liver seems to do a pretty good job at still working. And I think that comes down to the fact that we know that the liver can, like I said at the start, regenerate quite fast. So I think it can cope with all these cysts hanging around quite a remarkable organ. I don't know if that's answered your question, or if it was 5 tangents in one.
So, but the cysts only actually form on the bile ducts and not on any other ducts or structures within the liver.
That's correct. Interestingly, sometimes the cysts are not connected to these bile ducts in people. They're actually just sitting by themselves in the liver, and it's quite interesting to know how it goes from being, you know, a cell in this big tubular structure to then being isolated, being independent away from this structure. But that's more interesting for me as a biological point of view. And you know, some of the work we did maybe starts to look at how that happens, which I'm not talking about tonight. But yeah, the more we understand about these processes, the more we can try and intervene and lead the research forward.
Thank you for
Can I-
that question, Martin.
Can I just add to that? Yeah, Scott, I've just had a liver resection, and the whole of that left lobe of my liver, according to the surgeon in his words, was completely rigid with
Oh.
cysts. So I wonder whether there were cysts way beyond the bile ducts, or whether they were just too numerous. But to be completely rigid, I know a liver shouldn't look like that or feel like that. So that's, you know, it's quite interesting what it does do, what the liver is capable of doing.
Yeah, it's remarkable, and it's, you know, it's kind of, yeah, definitely a strong fit in the liver code that, you know, you can lose a lot of the liver and it'll grow back to its normal size. And something so
Hopefully without cysts.
yeah, hopefully without cysts. Yeah.
It's a big surgery to go through.
Yeah. Yeah.
Anyway, thank you for tonight. Really, really interesting.
Thank you for speaking.
I'm just looking quickly at the questions that are coming through in the chat. Laurie's asking if she can get a copy of the presentation. Wouldn't be able to share a copy of the presentation as such, but the recording will be made available, providing everything goes to plan. So we will have that. Someone's passing comment on their own medication. Jenny here's asking, and I don't think you'll be able to answer, what are the medications, number 1 and number 2? I think that might be a nudge, nudge, wink, wink, trade secrets. Unable to answer, Scott.
Well, not really my point, I guess, but it's more just that, you know, these aren't things that would be, you wouldn't be able to ask your physician for this, unfortunately, because they're not approved for human consumption or humans to take them. And it's a bunch of letters that probably don't mean much. Yeah.
John and Zoe were asking, saying that it sounds encouraging and the next steps relating to this research. I think you'd answered that earlier when you were chatting there with Raquel.
Yeah, so I guess we're now in communication with a pharmaceutical company who are doing, they're in a better position to actually try and drive this forward to get into clinical trials for people. So that's where we're going with that.
And someone asking about DNA genetics in polycystic kidney disease. So we have directed them to our website. It's not a question for tonight's presentation for yourself, Scott.
Okay.
Right, and we have Julie. And are you managing to see these on your screen, Scott?
I am, yes, yeah.
Yeah. Do you want to read through of Julie's and extract what you'd like there?
Sure.
She's mentioning a study that she'd participated in Yale. So we've actually got community members from across the pond tonight. So while you're reading through that, I'll scroll down.
Oh, I think I pulled out something that I think someone else has brought up, and it's you know, how common it is, or these processes for people who have, you know, if you have polycystic kidney disease where you have you know, cysts in your kidney and in your liver, or someone's mentioned that you get isolated liver cysts, so no kidney involvement at all, which is much rarer. Which is a really interesting point, because what we did in our research is we compared, we did have access to some patient tissue, so that was from our collaborators in the Netherlands, and they know the genetics, they know what the genes that were driving these cysts. And what we found was that for those, if you remember that TGFB1 molecule that I mentioned in the first part, and then the matrix receptors, I won't go through the presentation again, but we found that these were common things that occurred regardless of what the gene was that was causing that patient to have cyst. So that is really promising for us because it means that, you know, you don't have to separate people based on whether it's they've got polycystic kidneys and liver or they've got polycystic liver. We think a lot of these processes are the same. And interestingly, this might be even more important, is that we got some livers and kidneys from another rat model from our collaborators in Spain. And what we found again was that even in the kidney and the liver, these are very similar processes we think are happening. So it's kind of my ultimate goal is to get forward with somewhere where you don't have to, you know, take one medicine for your liver, one for your kidney. You can hopefully just take one that will combat both. We're not anywhere near there just yet, and I don't want to give anyone too much hope, but I think that, you know, the signs of the research that I've done so far are pointing that is possible. And I think that has answered quite a few people's questions in the chat.
It is, and it's Gillian there was, and Gillian, I'll say there's no such thing as a silly question. I usually ask those. And she's asking, is it likely that cilia and the chemical messengers and receptor mechanisms for cyst formation in PKD will be the same as in the liver?
Yeah, so essentially, yes. I think there's different parts to it, but I think the ultimate way that we can stop these from getting bigger and getting more numerous are going to be very similar processes, and that's really promising, I think.
Yeah, and Redmay is commenting, in medical science and research, will there be a cure for liver and kidney disease, or to reduce the cysts in the inside of both organs? I think, Redmay, that is why most of us get up in the morning. We always hold that hope that one day research will come to fruition and we see the end of PKD and PLD in itself.
Yeah, it's really interesting that we're getting a lot of those comments, because that's what I'm hoping to be able to go forward with, is to look at how similar these are, and I think they are very similar between both kidney and liver.
Someone is asking about polycystic liver disease. Does it only affect PKD patients and genes, etc.? Very little information available from your consultant. There is wider studies available which show that certain genes in PKD can impact on the liver, and you can have a standalone polycystic liver without PKD. So that is something, again, please visit our website, look at those educational seminars from previous, and you may find some of the answers there. Scott won't be able to answer a lot of the genetic questions.
Okay.
I think that's kind of covering. Are you scrolling? Is there anything catching? I think we're...
It's a lot of, I think people have the same kind of questions pop into their heads.
And Ruth there has commented just to say that she has, I think 13 years ago, had a transplant, and her hope is, would your research help reduce the need for transplants?
Yes, that's the ultimate goal. I assume you mean liver transplant as well? If not, but yeah, the ultimate game will be to intervene early. That's, you know, these are things that these cysts grow throughout, you know, the lifetime. So if you know you've got it, we need to try and find things that can stop them from getting too extreme, and yeah, tackle it early on.
Yep. And I don't know, we've got one here, I think it's Holmes, maybe the surname, is cilia missing in our livers?
Very interesting question. So we've looked at in the livers from the samples I got from our collaborators, and actually some samples we were getting from NHS. It varies a lot, even within the same samples. So from the same liver, there'll be regions where the cells do have cilia and some where they don't have any cilia. And yeah, we just used the way that we did it in our experiments was if we removed the cilia, we thought that we'd get cysts, and we were right. A lot of people have shown that in research. But yeah, it's an interesting question.
It's lovely to see some messages of thanks coming through, especially the fact that it's looking at a fairly rare disease, and it's a thank you for caring, which is a lovely. So we do appreciate giving your time tonight. It's really fantastic. I think, is it Catherine there's? Well, Catherine, are you wanting to join the chat? No. Okay, it was just I seen a hand get up, and that was you. Is there anyone perhaps in the audience who wishes to speak live just now? Julie's asking if there's any way she could get a copy of your paper. I don't know if it's free, perhaps for access, but we can certainly share our newsletter, that article that appeared in there with you. And someone, will there be a cure for PKD? Ultimately, that is an aim for us all.
Yeah.
And given the research that's already undergone, bearing in mind that I'm quite new to all of this information, there must be a lot of research that's already taken place. I would definitely look at what you suggested, the educational events. I mean, is that looking possible? Is it looking possible? I mean, what you've just said, Scott, is really positive, and probably the most positive bit of news I've heard about the whole process, or the whole medication side of things. I mean, has there been lots of movement to find a cure? I'm sure there's lots of work going on behind the scenes, but you know, is it something that you think that we're borderline finding a cure for, or is it completely underfunded? And
I think for the liver side, definitely there's not as much research there. The kidney research is definitely outweighing the liver by a lot. A lot of focus on kidney research, and you know that does make sense because of, you know, the kidney, the symptoms experienced on the kidney side are a lot more dramatic earlier on in life compared to the liver. Like I said, the liver can handle a lot. It seems to handle quite a lot of the cyst, but it can get to the point where it is too much. As far as I'm aware, I'm the only person purely focused on polycystic liver in the UK. I'm willing to be proven wrong if someone knows somebody or someone listening who does research here. Our collaborators, you know, we've got a few that are in Spain, Italy, Netherlands. That's because we're kind of dependent on them because they've been working on it for quite a while, and we're able to access their resources. But yeah, it's not, the liver is definitely, the research into polycystic liver is nowhere at the same pace.
Yeah, that's what I thought. So Scott, my thing to you then is please continue doing a great job, and we'll watch your career closely. I think everybody on this call.
Yeah, nice to hear. Thank you. Yeah, I mean, the ultimate aim is to try and get somewhere with it, right? So we'll keep at it.
Thank you.
Lots more messages of thanks coming through, and Mike has commented, for research, can a PKD kidney be used when a patient dies? Yes, you can, and PKD Charity actually fund a biobank in London. We do have the biobank there, which costs about £25,000. We do fund that for various tissue samples, etc. So that is available, and that is widely used for a lot of PKD research projects. So I'm just glancing down. Thanks. Artificial livers and kidneys may happen one day. At the moment, the artificial kidney, so to speak, is our dialysis machine. I think there may be at one stage something that can be implanted or portable, but potentially we will look for a cure as well. So that, I think, is everything for tonight. Just a final call if anyone wants to add a question. Final last chance. Nope. Thank you. What I will say on behalf of everyone, Scott, is a huge thank you.
Well, thank you.
I think you managed to get the right level there. Okay. Most people, I think, managed to keep up. I know that at some stages, I think my own brows were going. But we'll share the recording with you. It will likely be the early part of next week before that's available. We'll need to top and tail it, but certainly we'll get that escalated to you. We'll also, in that email, pop in the links for our educational events where you'll find them, some topics that may be interesting to yourselves, in particular our PLD ones that are there. I'll also pop a note on, say, how you can get the electronic copy of that newsletter, the front page news, that you can see that. And also just on that, it's a good point of, if you want to receive our newsletter, please sign up, because that's the type of things that you are hearing about. We do not sell your data to other companies. We do not share it, and any time you can unsubscribe. If you don't want to hear it from us, just switch us off. But that's certainly what is available to yourselves. I will ask, we usually do a formal survey about what you've gained from tonight for your well-being with living with PKD and PLD. I don't think it's appropriate tonight, but I'm sure we'd love to know what you thought of us as a charity supplying a research topic. Bit of a change for us. So doing that, and also if there's any comments you'd like to pass to Scott, we will certainly share that with them. And if you think we've got it wrong tonight, that, yeah, research, lovely to read about, I don't want to hear about it, let me know, because we'll work the programme to our community. So thank you. Enjoy the rest of your evenings. And if you have a burning question, do email myself. You will have my email, and I can forward it on to Scott. Right, good night. Thank you all.
Yeah.
Thank you.
Thank you.
Thank you.
Bye now.
Very good. Bye.
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 Thank you.
Bye.
Bye. Bye.
Bye.
Thank you.